We now proceed with the lecture of carbohydrate, which is the third macromolecule to be covered yet. So basically, this lecture is quite long. I have to break it down to two sections in terms of video. The first section is now been covered. It is based on the structure itself, that is overview on carbohydrate. And then we will look into the monosaccharide, a disaccharide, polysaccharide. Here I speak to you about the carbohydrate itself, where it has a ratio of CN and then water also multiplied by this N. You will see that compared to lipid, carbohydrate, the lipid has much more hydrogen rather than oxygen. That means it's in reduced form, and carbohydrate is more of oxidized form. And then it can be classified according to location, structure, and size. Location wise, you have starch found in plant, chloroplast, and then you have glycogen found in either liver or muscle. So it varies in terms of location based on the type of carbohydrate. Then you have structure as well as size. You can see on your right, you have glyceraldehyde as well as dihydroxy acetone having only 3 carbon. The carbohydrate monosaccharide can then increase size from 3 carbon to up to 7. And then the polysaccharide obviously can be much bigger in terms of number of carbon. So that's the difference in terms of size. Structure wise also, you can take the example given on the right, that the top one is an aldose and then the bottom one is a ketose. I give you overview on carbohydrate. It has a lot of hydroxy group, therefore polyhydroxy. Actually, this OH group connected to carbohydrate makes it very soluble in water. Solubility in water is determined whether it has a lot of OH group or charges. And then the carbohydrate is either aldehyde or ketone, meaning aldose or ketose. And then it is also having modified or derivatives of carbohydrate, which we will look in the second part of this lecture itself. In the second paragraph, it's given why it's been called carbohydrate, because initially they thought it's a carbon that is been having water molecule or hydrated, therefore the term carbohydrate came about. And then in the next slide, we will look into diversity of the chemical structure itself. In this slide, given the breakdown in terms of monomer up to polymer. The monomer is monosaccharide for carbohydrate, and it could be given in the form of example glucose, fructose, galactose, mannose. And these monosaccharides do not have glycosidic bond because they are not connected to any other monosaccharide. Disaccharide, on the other hand, is a combination of two monosaccharides, and it has one glycosidic bond. Example given here is maltose, where it is made up of 2 glucose and it is made up of alpha 1,4 glycosidic bond. So also varies in terms of the glycosidic bond, and also in terms of different disaccharide will have different monosaccharide combined in them. You have also sucrose and lactose. Oligosaccharide is not so popular, but has been referred here, it's up to 10 monosaccharide. Not important for you to actually remember it. You have also polysaccharide. You have starch, glycogen, cellulose. It is a combination of many monosaccharide and lots of glycosidic bonds to make up, for instance, in the making of glucose into becoming starch. You have amylose and amylopectin. You also have glycogen with much more branching than starch. as well as cellulose. It is made up of glucose as well. This slide is actually referring to that all the energy comes about from sun. So sun energy is absorbed by chlorophyll in green plants, and then this energy can be used to connect carbon dioxide to water and then form glucose eventually. And in the cells of a plant, as well as once the herbivores take up the plant in through food chain and then finally come to you, to humans, it is then digested and to form glucose in the cell, and cell oxidizes the glucose, and then you have reversal where you can form energy again in the form of ATP. So basically it's talking about how the energy in the carbohydrate can be harvested in to be utilized into becoming ATP. So now we cover monosaccharide, and then later we progress to disaccharide, polysaccharide, and also modified carbohydrates. So here monosaccharide, they are the simplest sugar where no glycosidic bond, and then a bit more on chemistry where it's 3 to 7 carbon with the uppermost carbon, carbon 1 put on top, and then also carbon 2, and then it goes on up to carbon 7 if the carbohydrate is having that many carbon. But basically it is arranged in vertical, and this is called acyclic structure. And in terms of whether it's aldose or ketose, if it is aldose, it has the carbonyl group at C1. If it is a ketose, the carbonyl group will be at C2 instead. And this is the way or manner of giving the structure for the monosaccharide. For this lecture, there's a lot of chemistry terms, and here in this slide you see the constitutional isomers. Constitutional isomers are those isomers that has the same number of carbon, same number of hydrogen, same number of oxygen, as been shown here for glyceraldehyde being the constitutional isomer for dihydroxyacetone. The only difference is one is a aldose while the other is a ketose. Similarly, for glucose as well as fructose, they are similar in terms of number of members, except being arranged in either to be aldehyde or ketone. And the glucose as well as fructose ends with the suffix of OSE. Unlike glyceraldehyde and dihydroxyacetone, still maintain in its original name itself. And then you have also the number of carbon, and also how to call them based on whether they are aldose or ketose. So based on number of carbon, if it's 3 carbon, it's referred as triose, and then 4 carbon tetrose, pentose, hexose, as well as heptose for 7 carbon. And then if you combine whether it is aldose or ketose, for instance, you can also refer them as aldotriose, meaning it's aldehyde and has 3 carbon, or ketohexose, it's a ketone but has 6 carbon part of its members. Then you also have more on the chemistry terms. For instance, the stereogenic centers for carbohydrate refers to the carbon connected to 4 different groups. Therefore, it will have stereoisomers. And then in the paragraph middle there, it's about how it can show the structure, the projection of Fischer. Later for acyclic is a Fischer projection. Then for cyclic we will see Howard projection, as well as the Bode and chair. Slowly we will learn all this. But in terms of the paragraph, the middle paragraph, in terms of how to draw the wedge line as well as the straight line and also the dash line we will see in the next slide. So here we learn about Fisher projection. Fisher projection can be as simple as shown here, where it is one-dimensional towards the plane of the slide here. Then it can be a bit more two-dimensional, okay, or altogether to be three-dimensional. In terms of one-dimensional, it is on the plane of the slide, so no difficulty in understanding. And then you have this wedge line. The wedge line is coming towards you, closer to you, compared to this dash line. Okay? The dash line is going further away from you. So I hope you can imagine that the OH here, as well as hydrogen, is coming towards you, nearer to you than the carbonyl group as well as CH2OH. And then this one is additionally, there's these straight lines on the plane of the slide, and then the wedge closer to you and dash line away from you. So this one more of 3D rather than these two here. And also the statement here given, do not cause the OH to be put here and the hydrogen to be put there, then it will be a different enantiomer altogether. If you put a mirror here, then you will have a different enantiomer image. So be careful on not doing that. So here we're going to talk about the family of D-aldoses, and then later we'll talk about the family of D-ketoses. So you have the carbonyl group, and then you add more carbon. If you already had 4 carbon as been shown here, there will be 2 stereogenic centers for the aldose. And if there is 2 stereogenic centers, in total there will be 4 stereoisomers. That is D-erythrose, L-erythrose, D-triose, there's also L-triose. In the next slide, we'll see all these 4 stereoisomers or enantiomers. So that's in terms of the aldotetrosis. Of course, if the stereogenic center number increases, there must be a way to determine how many stereoisomers. There's actually a formula, and that is shown in the next slide. So this is the formula utilized, where it's 2 power of n. The n represent the number of stereogenic centers. So if there is 2 stereogenic center for the aldotetrose, then these are all the 4 enantiomers relevant for this aldotetrose. So you have the difference because of the location of the OH, as you can see here. So there will be 4 enantiomers for this aldotetrose. Here put together all the family of D-aldoses, but you have to realize because it's family of D-aldoses, what is not shown is the L-aldoses. So if you have D-glyceraldehyde, do remember, because it has 1 stereogenic center there, there should have also L-glyceraldehyde. Similarly, for 2 stereogenic centers, total will be 4 enantiomers, that is D-erythrose, D-triose, L-erythrose, L-triose. And it goes on, this should be 4 or total 8, and then this one with 4 stereogenic centers, the total number should be 16 rather than what is been shown here, only 8. So the family of D-aldoses, the popular ones are ribose. You also have D-glucose, very, very popular. Mannose, galactose, also popular. Okay. So here spoken about the selected popular Aldo sugars. There you have ribose. Remember, this is the sugar found in the nucleic acid in RNA, and then once it's converted to deoxyribose, it will be found in DNA. Okay. Then you have the very, very popular one, the D-glucose. It's so much been utilized in your body, therefore it's referred as blood sugar. You also have others, galactose, mannose, and also where they come about in starch, cellulose, and other fruit pectins, and so on. And also been referred another chemistry term, that is epimer, uh, glucose and also mannose, only being different in terms of here, only different configuration on this location, and therefore these two are epimers among themselves. So just like the family of D-aldoses, we also look into the family of D-ketoses. Again, remember, it's only referred into D-ketoses. There's also L-ketoses. So by right, what is been given here should be double the number compared to just referring to D-ketoses. So you have from the very basic 3-carbon dihydroxyacetone, where ketoses have the carbonyl group at C2, and then you progress to further bigger number of carbon, and the popular one is D-fructose for D-ketoses family. Here we look into that in physiological condition, actually the acyclic structure of the monosaccharide is not favored. This is the acyclic structure. By right, it should be in cyclic manner, and here the joining of the cyclic structure takes place, and then it could be either 6-membered ring. It's not, because it's not referred as 6-carbon ring structure, because there's also oxygen, and this 6-membered ring structure is known as pyranose or pyran, and also have 5-membered ring, furanose or furan. Okay. So this is the referred as acyclic or cyclic, and as you can see here, the arrows more pointing towards the right, meaning it will exist as cyclic rather than acyclic most of the time. And lastly, also in terms of Hemi acetal, once the structure joins here, it will form a Hemi acetal structure at this location. Here shown the formation of hemiacetal as well as hemiketal. In order to form hemiacetal, you need an aldehyde, and then you also need the functional group or alcohol. Eventually they react and then form hemiacetal. And then to form hemiketal, you need ketone rather than aldehyde to link with alcohol. In terms of the hemiacetal as well as hemiketal, they will form a stereogenic center whereby the carbon is linked to 4 different groups. Although this is oxygen, but it's actually considering both together as one different group. So here we look into the cyclation. So you have something which is acyclic, and then because of these two electron there, the carbon can form interaction. And once it forms the interaction, there is two possibilities, either alpha anomer or beta anomer. So what's the difference between these two? They call it distereomer, and that's been defined by this term here on how to know which is alpha anomer and which is beta anomer. The alpha anomer has been written here is basically the OH is far away or what do you call position compared to the C5. So that's because it's far away, therefore it is referred as alpha anomer. Then the beta anomer, the OH is at cis position to C5, nearer to C5, and therefore it is referred as beta anomer. So here given more facts, that is after the glucose acyclic become cyclic. So you have the, just now we talk about Fischer projection. For acyclic, once it form cyclic, as in the glucose just now, you will now talk about something referred as Harvard projection. And this is specifically in terms of how the cyclic structure can be seen in terms of its positioning. And then later you will see we will also talk about chair and boat projection, which is the best positioning of the structure. And both the alpha beta D-glucose are considered distereomers because of the two types of positioning of the OH. And then if it's in acyclic form, it will have 4 stereogenic center. But as soon as it forms the anomer, the Hemi Acetal, it will have 5 stereogenic centers. Okay? And lastly, talking about the positioning of alpha anomer to be trans as well as beta anomer to be cis, which I already described to you. This slide basically describe in terms of chair or boat configuration, although here if you look at it, you should be able to see a kind of chair projection, uh, not a common chair, a chair found near the pitch side. So the glucose will take this chair configuration much better than the Harvard projection because this one has the least amount of strain, energy strain on its interactions. That's why this is much more preferred than the Harvard projection. Just to highlight further, these wedges here, okay, these wedges are actually coming towards you compared to these straight lines which are going further away from you. I hope you can see the chair position or chair structure because when I give this lecture in the classroom, when I ask whether you can see chair or boat, some students cannot even see the chair structure at all. But here both configuration is not being shown at all. Just now we looked at glucose in terms of forming furanose ring structure, but here we are going to look into furanose ring structure formation, and this one looking into aldose, and later we will see ketose. So in terms of aldose, you will have the carbonyl group there at C1 interacting with the C4, and this is the acyclic form where it can, once it forms the cyclic structure, it can be in two forms, either alpha anomer or the beta anomer. Okay? Has already been described to you. Alpha anomer, the OH is at the bottom, while beta anomer, the OH is at the top. Also want to highlight here the Harvard projection. You can see this is Harvard projection where the wedges are closer to you and the other line is far away from you. I hope you can see the Harvard projection in this slide. So we already looked at how Aldo hexose can form the cyclic structure. In this slide, we look at how keto hexose will form the cyclic structure from acyclic. So you have the C2 carbon now with the carbonyl group will react to the C5, and then it will form either alpha anomer or beta anomer. Now we move to something else, which is again much more complicated and what we already learned. So you have to understand this lecture is quite heavy in terms of a lot of chemistry, but that's how it is in terms of carbohydrate. So we now move into understanding mutarotation. So what is mutarotation? So you have the acyclic structure of glucose, and then it can become cyclic. When it becomes cyclic, it could be either alpha anomer or beta anomer, and in terms of the quantity, it differs. It will be in alpha anomer 37%, while in beta anomer 63%, while in acyclic, which is less than 1%, so it's trace. So in terms of the total, 100%, but it will be in equilibrium in these three forms. And because it's in these three forms, it is possible to have chemical reaction due to these three forms. For instance, there is reactions because of the carbonyl group. There could be also reaction because of this anomer, the anomeric carbon, either the OH or the Hemi acetal itself. You also have reaction due to the OH, which is not in the anomeric carbon itself. So these are all the reaction which we will see in the next slide. So this is continuation from the previous slide just now about the formation of cyclic and acyclic structures. When it forms these two different structures, it will be able to have few reactions which are confined to these two structures. The reaction could be hemi-acetal, that means involving the hemi-acetal carbon. It could also be reaction of the hydroxy group, either the hydroxy group belonging to the anomeric carbon or the non-anomeric carbon. It is also involving the acyclic, which is in trace level, the carbonyl group. So these are the three types of reaction, and we will see in the next slides to come the examples of these three reactions. So here given the example of reaction at the hemi-acetal group, whereby you have this group here, this carbon, when in the presence of methanol and hydrochloric acid, the reaction will be very vigorous to this group here, and what will be end result is two glycoside will be formed. One is alpha glycoside and the other one is beta glycoside. So shown here in this photograph, actually if you go and Google or you go into the YouTube, you will find a video that actually shows this formation of black snake once the sugar been added with the hydrochloric acid as well as uh, the methanol. It will be a black snake, as you can see here, coming out in a vigorous form. And this is the to know whether the sugar is reduced sugar or non-reduced sugar. The reduced sugar or reducing sugar has ability to reduce the indicators has been given here. Toluene region, which has a argentum ion, will then be reduced to become argentum, and there is a silver color for the element argentum. So that's the indicator there is reducing sugar. You also have Benedict and Felling, which has copper 2 plus will be converted to copper 1 plus, and therefore there will be change in color, blue to brick red solid. And I show there in the photo there how the brick red solid looks like. The ability of the reducing sugar to reduce these reagents comes about because of the carbonyl group due to muta rotation from cyclic to acyclic. So here shown the reaction itself in terms of how reducing sugars can convert argentum indicator to become silver argentum, copper in Felling as well as Benedict from copper 2 plus to copper 1 plus, therefore change into color into solid brick color. And also additionally there is also bromine gas which can become bromine ion, so there also will have changes in color. Instead of the indicators undergoing changes, you can also notice the reducing sugar will be changed from carbonyl to carboxylic group. So from what was glucose, it will become gluconic acid. So you have to realize that the possibility of reducing sugar only comes about if there is Hemi acetal. So this glucopyranose is a reducing sugar because of this carbon, which is anomeric carbon, can mutarotate to be carbonyl group and therefore react as a reducing sugar. Similarly, the alpha D glucopyranose, tetramethyl alpha D glucopyranose, also have the anomeric carbon and therefore is hemiacetal. Therefore, it can reveal the carbonyl group when it become acyclic. The one that is not reducing sugar or non-reducing sugar is this methyl alpha D pyranoside, where it is acetal, not hemiacetal. When it's acetal, it cannot mutarotate and therefore it cannot become a reducing sugar. So if the glycosides such as acetal, they are not in equilibrium with acyclic aldehyde, therefore they can be confirmed to be non-reducing sugar.